Hi, so now finally the valedictory session. So before we start the final talk by Professor Nanjindya, we have the pleasurable duty of handing out the certificates for participation, and for that I have invited Professor Rahul Siddharthan, who's in charge of the IMSS Center for Disease Modeling and also the Computational Biology Project, which is actually providing the bulk of the funding for this workshop. So big hand to Rahul. So he'll be handing out the certificates of participation. So first, so I'll do it in alphabetical order. Abhinav Kanan from ISA Pune. Akshay Varu from Manipal Institute of Regenerative Medicine. Archana Naik from ISA Pune. Ms. Vairavi from Stella Maris College, Chennai. Ms. Divyashri from Sri Ramachandra Institute of Higher Education and Research Center. Mr. Gurjot Singh Sawhney from ISA Pune. Ms. Kangaban Limthoin Gambi from IISc Bangalore. I'm sorry, I really messed up your name, didn't I? Mr. Kulkarni Gopal Venkatesh from ISA Thiruvananthapuram. Mr. Mohammad Akhtar Ul Kareem from Institute of Chemical Technology, Mumbai. Mr. Nimal Achish Kanan from NICER Bhubaneswar. Mr. Nitin K.A. from ISA Thiruvananthapuram. Mr. Sarthak Saini from ISA Pune. Ms. Shaurya from NICER Bhubaneswar. Mr. Siddharth T. from ISA Mohali. Vaishnavi Dondiram Mugadio from ISA Pune. Sorry, I'm mistaken. Okay, I'll just tell her that I'll correct it later. Sorry, there was a small mistake there. I'll correct it later. Okay, now the IMSC people. Ananta Datta. Aniruddha N. Ishitwa Gupta. Oh, absent. Sorry? No, Ishitwa. Yeah. Anuran Pal. Absent. Okay. Kalyani Murali. Roni Saiba. Mrinal Kanti Paul. Ah, there he is. Onuran Pal. So yeah, so we just have Ishadwas left. So we'll hand it over. You can probably hand it over also, right? Ishita is working with you. Yeah. Right. Okay. So thanks so much, Rahul. Yeah. All right. So now, without further ado, let me invite Professor Nanjindya for his final valedictory talk.
Should I begin? Thank you once again, Chitabhra, and thanks to all of you for having stayed on, even though the temptation of wanting to go home or at the least to see Chennai after you received your certificates must have been quite strong. So thank you. As you will see, what I am going to say today really is a continuation of what I said yesterday. It's the same theme that I'm going to pick up, which is how to look at development from an evolutionary point of view and how to embed development into evolutionary thinking generally. And these are really pretty much the same subject as I was trying to tell you yesterday. I don't know how much I succeeded, but I shall reiterate that in what I'm going to present today. So one can see the problem of evolution really as whatever that number is, 100 million different problems. So there are these entities defined in some form or the other, which we call species, and there are 100 million odd of them. They seem to be 100 million solutions to some problem. What that problem was in the past and what that problem is remains a matter of some contention, but they certainly seem to be solutions. And one goal of evolutionary theory is to try and see whether there are general principles to which there are these 100 million odd solutions. So the solutions are in the form of not only their visual appearance, which of course is the first thing that strikes one about them, it is about those that we can see with the eye, but also about their behavior, about the way they interrelate with each other, and something which has struck everyone who looks at the living world, at the way in which they seem to fit in a certain manner to their circumstances of life. In other words, to this phenomenon called adaptation, which is believed to be the signature of living creatures. So this seems to be a problem worth tackling. At the same time, if the problem has 100 million possible outcomes, it also appears rather forbidding. If you look at development, however, it appears, and here I mean multicellular development, it appears as if those 100 million problems really collapse into maybe 30, 40, 50 problems. This thing says 35, but that depends on how you do the counting. So these are the major forms of multicellular development, which over the years biologists have classified into these broad categories called phyla, and the simplest sort of colloquial equivalent of phylum would be lifestyle. So, there are 35 broad lifestyles, and part of the lifestyle of course is incorporated in the form itself, form and structure itself. 35 ways of making a living according to this count. Now, this does not take into account plant types, but they are not too many either. The number of plant phyla is believed to be maybe between 10 and 20. So, let us say 15, that still gives you 50, and if you say what the hell, let us double it and make it 100, that is still a tractable number of solutions to some other problem now. And this number of course is much smaller than the number of solutions, many orders of magnitude smaller than the number of solutions to the evolutionary problem in the way that we posed it. So, one hope is that by tackling this maybe more tractable problem, we might get some insights into how that other problem might be addressed. The evolutionary problem, and by that I mean specifically now the problem of species numbers, automatically carries with it the problem of how you distinguish one species from another, and that distinction is based on many features, the most critical of which according to biologists is the inability of different species to exchange genes with one another. Species are within themselves reproductive islands. Now, if that feature is removed, it is not very clear that there are 100 million species either. In other words, if you say I decide to call everything that looks more or less the same to me and lives more or less the same kind of life, eats more or less the same kind of food, etc., etc., then that number collapses to a very small number. So, it is clear that this business of what you might call genetic barriers goes part of the way in explaining why you have such a huge species count. Keep that in mind. So, if the genetic barriers for whatever reason are ignorable, then the number collapses to a smaller number, and this, as you may remember from yesterday's talk, was one of the themes going around in what I was trying to present, which is that genes are very important, but do not try to make them more important than they are in trying to understand biology. Good. A few other general features about living systems and biology which any evolutionary explanation has to address. One is, unlike this, unlike the impression given by this picture, living systems are not frozen snapshots in time. That is the way we look at them usually, of course, for reasons of convenience. So, we think of the word frog as meaning something which is of a certain nature, human being for example, any one of us, etc., etc., whereas all of us, all living creatures really are entities that exhibit order, structure, form, characteristic behaviors, etc., which keep changing in time. And this changing aspect is embodied in the life cycle, and the word cycle here also tells you something which is very useful, which is that not only do living creatures change, but it is this pattern of change that seems to repeat itself from one generation to the other. So, there is some kind of periodicity implicit in the notion of living creature itself, which of course we associated, which is usually associated with reproduction per se. So, here you see a viral life cycle which essentially depends on some other living creature. You also see a bacterial life cycle illustrated this way. You have a life cycle of one of the creatures that you listened to, heard about yesterday, these cellular slime molds, and this shows you that within this one creature there can be more than one kind of life cycle. So, it is the same creature, but exhibits three different temporal patterns which recur generation to generation. One of them is the vegetative cycle, the other is the cycle that we spent some time on, aggregation, forming spores and stalks and then reproducing, and third is the sexual cycle, which we did not talk about at all, which is also an independent life cycle. Then there are these life cycles which are perhaps the most familiar to us because we form part of them, where you have a single cell meeting another single cell, the two nuclei getting together, and then going through a whole series of cell divisions, movements, specific attachments, and ultimately what we call differentiation, which is shaping, which is some kind of architectural enterprise, which gives recognizable three-dimensional form and shape to the organism. And you can see this in frogs, you can see this in animals, you can see this in plants and so forth. One important point to keep in mind here, which was distinct, which was not a central feature of the previous two life cycles, is that here the life cycle as illustrated here on this screen is not an entity by itself, it is not an autonomous entity by itself, because the starting off point, at least the conventionally designated starting off point, which is fertilization, requires the coming together of two individuals, which is really two life cycles. So there is no such thing as a life cycle by itself which gives rise to another life cycle in multicellular creatures. There are two life cycles which in some way combine and give rise to a third life cycle, worth keeping in mind. If you look at older discussions of evolution, and I am speaking here even of pre-Darwinian discussions of evolution, and these extended beyond Darwin's day, in fact even in the literature that we look at these days, quite often the words development and evolution were used interchangeably. So what we call evolution today was often in the literature described as development. In other words, the word development was used to describe it, and similarly it is not uncommon to come across in the literature phrases like the evolution of an embryo into an adult, by which they mean of course the development. So this interchangeability in language already showed that there were many features in common between these two, at least superficially common between these two, that people try to capture in words. One feature of development that was realized very early is that it exhibits a spatio-temporal pattern of reproducibility, and that has been expressed, I think, first by Cohen and Robertson, but perhaps by others as well. That has been expressed in terms of the phrase developing embryos have within them clocks and maps. So the cells within a developing embryo do what they do depending on what time it is, so to speak, depending on their assessment of the time, and depending on where they are within the embryo in relation to the rest of the embryo. So they have both a temporal coordinate that they access and respond to, and a spatial coordinate that they access and respond to. There are other features that the behavior of a cell depends on, and these are listed here. I have already mentioned its position and time. In addition, importantly, the behavior of a cell depends on its past history. In other words, its developmental history during that life cycle, and it depends crucially on its environment, which in this case, other than the physical environment, comprises the environment made up by its neighboring cells. So where the cell is, what time it is, its past history, and the interactions that it has are the inputs that go into a cell. If you think about it a bit, these are precisely the sorts of inputs that go into deciding the evolutionary fate of any particular species. And I am sorry, so I will come back to that. 1 second. Whoops. Yeah. So these are precisely the kinds of things that impinge on the evolutionary fate of any given species. And when you think of it, you can jot down a few commonalities which strike the eye when you think of explanatory principles behind development and evolution. And of course, you can also think of situations in which these principles do not seem to overlap. So here I have listed a few. The most commonality of course is that both of them involve, both development, multicellular development and evolution involve what appear to be dramatic changes. So, on the face of it there is something close to a miracle happening, and if there is any of you here in the audience who hasn't seen development taking place with their own eyes, please do so. It's the easiest thing in the world to do. Now the rains are about to start and you can collect frog eggs anywhere. All you need to do is to put some eggs in a moist environment and look at them through a microscope. It's an incredible sight to follow what happens, or fish eggs for that matter. Okay, so the dramatic changes both in development and in evolution. In evolution of course much of the time you don't see really dramatic changes, at least not in form and shape at the time scale of a human life, but nonetheless we have begun to appreciate over the past 100 odd years that such changes have taken place. Then it is fairly clear that both development and evolution are kinds of things that are exhibited, kinds of phenomena that are exhibited by matter in the broadest sense of the term. So, the properties of matter must lie behind any explanations that you offer for them. In other words, the ordinary laws of physics and chemistry must play a role, of what kind we do not know yet, but must play a role in deciding on how evolution takes place and how development takes place. Both of them involve gene based inheritance, by which I mean that in development the cell that a cell gives rise to depends on the genes of the mother cell, and what it gives rise to, in other words the outcomes that the daughters of that cell has, also depend on gene based inheritance. This is of course taken to be trivially true about evolution. As I said at the beginning, the defining mark of the difference between two species is believed to be their genetic makeup. The environment is important, is central to behavior, meaning to how differentiation takes place in one case and evolution takes place in the other case. And most importantly, something whose depth and whose explanatory power is only now becoming apparent to us now. Chance, both in the sense of what you might call random events, meaning events which can occur many times but each time in an unpredictable fashion. Chance, both in the sense of random events and in the sense of contingencies, which means not necessarily something which happens many times, but something which happened to happen on a given occasion, and there is no compelling reason for you to think of an immediately causal explanation for it. In other words, you can walk from here back to your hostel any number of times, and once you find that you trip and slip, that is a contingency which occurred then. So, it does not mean that there is a certain fixed probability at which that occurs on every walk, but it just so happened one time, and it happened to happen because it rained just before and there was a puddle where you did not expect it, etc., etc., etc. So, contingencies play an important part in evolution, we have learned now. Turns out, so do they in development, though perhaps not in as obvious a fashion. Randomness, which is the second aspect of chance, plays a role in both, and during this meeting itself, including during the presentations that many of you gave during this day, earlier today, you saw what a central feature of development noise was, and so it is in that sense that one uses the word randomness in development. In the case of evolution, of course, randomness is central to the Darwinian picture of evolution, in the sense that mutations are believed to occur at random, and recombination, which is another very important feature of inheritance, is also believed to occur at random, genetic recombination. So, there are these similarities. The dissimilarities are also fairly obvious. The time scales over which they occur are hugely different, hugely different. To get perceptible evolutionary change might need hundreds of thousands of generations, whereas here you are talking of a time scale which is a fraction of a generation, so within a single life cycle. Then there is this difference which keeps cropping up every now and then between genetics, which lies behind evolution, genetic change, and this sort of pop word that keeps getting used all the time, epigenetics, which is behind developmental change, which is to say how it can be that the same genetic makeup can actually give rise to very many different outcomes which are inheritable. So, the point is not simply one of the outcomes being different, but of the outcomes being inheritable. And here of course one uses the word heritable or inheritable in the sense of heritable within a lifetime. So, from one cell through mitotic generations, not meiotic generations, which is genetic inheritance. Development is repeated, so same thing happens again and again and again, and that in fact is the bedrock on which theories of development are built, their reproducibility, their reliability, whereas evolution is believed to be, and as far as we know is, entirely unique. It is a once forever, if you wish, occurrence which is taking place. So, keep both of those in mind. Already from Darwin's day, it has been realized that development has a great deal to tell us about evolution. Darwin devoted a whole chapter of his famous book to this subject, and in fact there are lots of hidden nuggets within that book which I strongly recommend all of you to read in case you have not done so. It is a very readable book too, by the way, The Origin of Species. There are all sorts of hidden nuggets within that chapter which people keep repeating these days as if it is something new, but there is much deep thought which went into writing the chapter as with the rest of the book. But the basic point Darwin makes, which of course has been repeated in textbooks ever since, is the analog of this kind of illustration, which is not Darwin's but came much after him, slightly after him. And the illustration basically makes the point that if you look at multicellular development, and here you have a number of vertebrates, fish, salamander, whatever, turtle, chicken, frog, etc., including human. If you look at multicellular development in a number of vertebrates, it appears that you see some kind of a branching pattern where all of these multicellular creatures to begin with are more or less indistinguishable externally, and differences between them start to appear serially in the course of time. So, some differences to begin with, more differences later, still more differences later, and so on and so forth. And this sort of parallel, which Darwin of course went into in great depth, and which present day biology has continued to follow up by its studies of gene action in different organisms and of homologous genes in different organisms. These studies have only served to confirm the point that this extraordinary similarity in resemblance must mean that there are similar underlying principles behind the development in all of these forms. The implementation of those principles has varied between forms due to reasons that we still need to follow up. In fact, this is a direct quote from Darwin. Embryology rises greatly in interest when we thus look at the embryo not as a picture more or less obscured of the common parent form. When we look at the embryo as a picture more or less obscured of the common parent form of each great class of animals. So, the idea being, of course, this is the central theme of his book, that species have diversified from a small number of common ancestors, eventually going back to a single common ancestor. Coming now to theories of development. There have been two broad classes of theories, and under different names the same classes of theories continue to be with us today. In some ways this is reassuring, shows that people had long ago realized what the basic distinctive points were in different kinds of development. In some ways it also shows that in terms of fundamental theoretical insights, we may not have moved all that far from what people had thought of long ago. So, these two theories go under various names. I have used the oldest such name, preformation and epigenesis, not epigenetics, epigenesis, which is a very old word. These distinctions have been named differently in the course of time. So, that distinction between preformation and epigenesis is also described sometimes as a difference between a determinist and a contingence based model of development, or between mosaic and regulative development. That used to be a very popular term in embryology textbooks and developmental biology books when I was a student. And of course, in a sense, it also mirrors the difference between the centrality of genes to developmental change and the centrality of the environment to developmental change. So what is this difference? The pre-formationist theory originally held that the main thing that happened during multicellular development was growth, an increase in size, which of course is a feature of development, no one can deny that. But pushing backwards and seeing how babies, at least at birth, really look pretty much like adults, except that their proportioning of body parts is very different. Generalizing from this observation, the pre-formationist theory held that if you go sufficiently back, what you have is a miniature baby, miniature to the extent of being invisible to the eye, a miniature baby hidden somewhere. Now, hidden where, this wasn't clear. In some theories of pre-formationist development, it was hidden within the sperm, in which case fertilization, which was realized very early as being essential for development, fertilization was taken to be an event in which this incipient baby actually got the breath of life, so to speak, from the egg. So the egg served to activate the baby and then make it proceed towards development. The complementary theory was that this tiny baby, it was even given a name called homunculus, this tiny baby was actually within the egg and the sperm performed this activatory function. In either case, it was a repetition. Of course, like all theories which go back to explaining something that happens now in terms of something that happened earlier, that was there earlier, this begs the question of where that came from. But within themselves, these theories were satisfactory because they said, these pre-formationist theories, because they said that everything was cyclic. So you had some explanation behind this cyclic phenomenon, which you didn't have at hand at the moment, but once you got that explanation, the problem of development was solved, because development per se was simply growth. The counter theory was this theory involving epigenesis, and epigenesis really means some kind of unfolding, appearance of form where there is no form, and appearance based on things that happen within this thing that is developing. And the true originator of this epigenesis theory of development is really Aristotle, because he says very clearly, based on his observations, that form arises de novo during development. It is not there earlier, but appears in the course of development. Therefore, it has to be a phenomenon which you can describe by the word epigenesis. But the modern theory of epigenesis is usually ascribed to William Harvey, whom we know better as the discoverer of the circulation of blood. He was also an anatomist and a surgeon, and he turns out carried out a large number of dissections of animal embryos and concluded on the basis of his observations that what was born was not there in any form earlier. In fact, what was there earlier in the embryo was a shapeless mass, indistinguishable in terms of its parts, which later got form and structure in the course of development. So, epigenesis. This, as I said, this distinction also falls under different heads: a determinist versus a contingent plan, mosaic, which is the earlier pre-formationist idea, versus regulative, which has a more particular meaning, which we don't need to go into now. And today we would look upon it in terms of the following two points of view. One, saying that genes are sufficient to guarantee how development takes place. I used this description yesterday as well. Genes contain a program in the sense of a computer program. Genes contain a program for development, versus the point of view which says genes set up a groundwork on which development occurs based on what happens later. And what happens later in our case is going to be interactions between cells and all the other things that I listed earlier. Broadly, an interaction between the unit you are looking at, which is the cell, and its environment. Experimentally in the history of development, and this is a common feature in many cases in biology, it appeared that you could come up with convincing results which pointed to either explanation. And the classic example in this case consists of the experiments of Rooks and Driesch, both of whom asked the question, if instead of a normal embryo you take half an embryo, what will it give rise to? Rooks's experiment, which is illustrated in the top half of this picture, involved killing one of the two cells following the first division of the fertilized zygote, and he found sure enough that the second cell, the cell which was not touched, gave rise to a half frog, actually not a half frog in this case, but something which looks like a half larval tadpole. It dies, it does not go on further to development, but nonetheless very clearly half the embryo, half a recognizable frog embryo. So the immediate conclusion was the half that had been killed contained within it factors for making the left half of the tadpole. The right half, which was untouched, was there to make the right half, which it did autonomously, the left half not being there. Driesch's experiment, and this experiment has been carried out by many people. Again, it's a fun experiment to do if you have access to very simple tools and a microscope. Can be done with frogs as well, but Driesch first did it with the sea urchin, which is illustrated here, which is he separated these two daughter cells from the first cell division, didn't simply kill one of them and let it be, and now it turned out that each daughter half gave rise to a completely normally proportioned adult. In other words, an adult without any missing parts. Here again too, it's really the larval form that you see first, but later the adult sea urchin also develops. Now the picture shows only the larval form, the adult form also develops similarly. So you see immediately that the conclusion from this was that each half contained within it the potentiality to give rise to a whole. Today of course we know that this is a seeming paradox which has been resolved, and the resolution comes in the nature of the experiment. Rooks imposed a mechanical barrier to the normal regulative capacity of the adult, whereas Driesch did not, and that made the difference. Already these two experiments showed that there was some level of autonomy possible in development, and the nature of that autonomy was shown in numerous experiments, none better to my mind than this one by Hemmerling on blue-green alga called acetabularia. This is a single cell, so it's not a multicellular organism at all. This is a single cell, the one that you see sketched on the screen, which has a recognizable morphology with what you would call body parts, except that these are a body within a single cell. And the recognizable difference in morphology comes from pigmentation, and also comes from this thing on the top called a cap. And you can see in this picture here, at least the species that Hemmerling worked with have two distinct kinds of caps. And his experiment was extremely elegant, and it was based on the fact that this blue-green alga is capable of regenerating a cap if it is cut off. So you cut off a cap, a new cap regenerates. Hemmerling showed that the form of cap which regenerated depend on where the nucleus came from. The nucleus, it turns out, it's in the cell of course, but it's at the opposite end of the cell, at the bottom, in this place called the rhizoid, which is a kind of holdfast with which the cell attaches itself to the substrate. And if you transplant a nucleus from another species, from Mediterranea to Crenulata or the other way around, and then perform this amputation experiment, the newly regenerated cap resembles not the cap of the host, but the cap of the species from which you took the nucleus. So the centrality of nucleus to form, which of course is something that we by and large continue to hold today, believe in today, was really first convincingly established by Hemmerling. Then there was a series of experiments done by a person called Volterek, which anticipated experiments which many of you have no doubt heard about, and as I was told yesterday, these were discussed in this meeting earlier. These other experiments that I refer to were done by Waddington, and I'll make a mention of them later. Volterek carried out these same experiments before, and what were the experiments? He looked at a morphological feature in a small freshwater in a crustacean, something called Daphnia. This is a Daphnia-like species called Hyalodaphnia. And the feature he looked at here was the head, which you see demarcated. So that's the portion of the body between B and C. You can recognize it because it looks like a head and it's got an eye there, eye spot as it's sometimes called. And Walter Eck first showed that the same Daphnia species, when raised in different environments, had markedly different forms, in particular markedly different heads. So the same species. And what you see on this graph is on the y-axis a change in head shape, and he quantifies it in terms of the size of the head relative to the total size of the body. And what you see on the x-axis is the quality of the food supply given to this creature. It usually feeds on small algae, essentially anything smaller than itself. It can even feed on bacteria and other things. As you can see, and this refers here to 3 different sources of Daphnia, the 3 different curves, each of which show how the head shape changes qualitatively, how the head shape changes quantitatively as a function of the amount of nutrition given to you. So basically, this head shape in the adult water flea or adult Daphnia is not an invariant property, but rather is an environment dependent property. That's the first point that he makes. The second point that he makes is that these variations that you get, which are purely environment dependent, are as significant as the variations that you get in naturally occurring distinct species. So these are a series of different isolates made by Walter Eck which show these different head morphologies. Second point. So the first point was that the shape, the size depends on nutrition quality. Second point is that the differences that you get mirror differences between naturally occurring species. The third point that he makes is that by repeated, you asking me?
He's saying he needs to get the charging connection to the
This is not my laptop.
but the laptop.
Yeah. Is that okay? So I repeat, the first point that he makes is of significant variation. The second point that he makes is that this variation, which is environment directed, mirrors the variation that you see in naturally occurring species. The third point, which is the most significant, is that after carrying out long generations of selection on these environment induced variations, Walter Eck is able to get a line of Daphnia which breeds true for this new variation. So there's some way in which, and I'm going to use a word now which you have heard previously in this program, I'm sure, there's some way in which Walter Eck is able to assimilate this variated form of Daphnia into the germ line so that it breeds true. Now, the question obviously is what's going on, and we'll see in a moment what's going on, which is an explanation which eventually Waddington gave. But when it was first found out, or at least first claimed by Walter Eck, it was fairly universally disbelieved because people said you can't get inheritance this way. This was already long past Darwin and past Mendel as well. People said you can't get this kind of inheritance. Nonetheless, not only could Walter Eck demonstrate this, but he said that this shows 2 things. One, you can get natural variation. That natural variation is restricted within species specific boundaries. Those boundaries can be exceeded, those boundaries can be broken by means of environmental stimuli. Having been broken, you can carry out an experimental program in which the boundary seems to be transgressed permanently by this form of selective breeding. The word that Waddington was to use later to describe this phenomenon, which is a word which has now become common in the literature, is canalization. And Waddington's description of the parallel phenomenon that he demonstrated, and I will show you a cartoon later, Waddington's description was to say that normally what we think of as species specific forms are forms that, as it were, flow along a given channel or along a given canal, do not transgress the boundaries of that canal, but they can be made to transgress those boundaries by means of environmental stimuli. And if that happens, there are certain conditions under which you can make them transgress those boundaries permanently. What those conditions are, we have not looked at as yet. The answer, it turns out, involves entirely conventional Darwinian selection. In this case, not natural selection, because it's the experimentalist who is applying the selection, but nonetheless perfectly conventional Darwinian selection. We'll come to that in a moment, but before I do so, whoops, sorry. No, I'm going in the wrong direction, I think. Yeah. Before I do so, just to hammer home this point. So there are two things Waddington, and it turns out at about the same time as him, Schmalhausen, there are two things that these two people pointed out natural selection was capable of. One, which is in leading to the evolution of a particular end point, so to speak, particular form, particular shape, particular behavior, particular morph, as you would call it. That's a standard way in which you look at evolution. There's something which evolves, and you have that thing in your mind as a given entity. But secondly, they pointed out that what evolves is not simply that form or shape, but that form or shape in a reliable fashion. So what involves is not just the mean outcome, but also the noise about that outcome. And this is the first time in developmental biology that emphasis was put on the role of phenotypic variation, noise in a general sense, as something which would also be looked at as an evolutionary product, and as something on which selection could act, in this case of course in the lab, but in principle on which natural selection could act too. So what Walter X experiment had done, it turned out now with hindsight, is to have reduced the noise, which was a natural concomitant of development in a new direction with a new head size. The experiment had succeeded in reducing that noise and canalizing or channeling development in a new direction. So selection can act on the mean of a trait as well as on the variance of the trait. Now this is something very interesting, and these ideas were picked up much later. Sorry, you had a question? Yeah.
As far as I understood, like idea of canalization is associated with the robustness of the developmental regime or the life history traits that basically guides the.
Right.
Now the idea of noise in it itself basically reduces that robustness.
Right.
So how is that basically canalized in that sense?
Well, these are two ways of describing the same thing, isn't it? Noisy development, meaning uncanalized development, meaning development which is not robust. On the other hand, if you get development with noise suppressed, by definition that development is robust and channelized.
So when generally the idea of canalized is like introduced, it is introduced in a sense that life history trait in general are like robust in a sense that they can no more be the selection pressure if
they can tolerate environmental or genetic perturbations strongly.
Yes. So now when we are basically doing selection or because of environment there is some change happening, then this idea of canalization itself basically kind of breaks down. I don't
Yes, exactly. That's exactly what Walter X said. I mean, he didn't say that, but that's exactly what he showed.
So basically we are saying that
canalization was broken down, the normal channel boundaries were transgressed, and now because of selection, canalization was again made robust, but in a different direction.
Okay, okay. Another question that
So
was.
think of it in terms of a water flow along one channel, overflowing the banks somehow, and then flowing reliably along a different channel.
Oh, yeah, I get it. So another question that was driven by like the initial thought of when we were talking about even if the genetic structure is same, the environmental variations itself are enough to basically guide the evolution, and they can be inherited.
I didn't say that. No, I didn't say that at all. I said that in this experiment, this happened. The experiment consisted of two parts. One, to use the environment to invoke changes, to lead to changes. Secondly, to channelize these changes in a new direction, but that required selection.
So, no, initially when we were talking about that, it's not just genetic structure that we should look at.
Yes,
Environment
in
is
a certain
also,
sense. Yes,
yeah,
yes.
environment can also guide the variations and then can guide the selection.
In a
the
certain sense.
evolution.
In a certain sense. Yes.
Yeah, especially also in this experiment where basically for certain duration of time, just the phenotypic plasticity, before the genetic assimilation has happened, there is some time when the environmental trait is being inherited, and after.
You have to you have to be careful there. The trait is being inherited only because the environment is your new environment now. It's not a trait that breeds true. Okay, so let me repeat the experiment. Think of nutrition. Let's say that when the nutrition is poor, the head of Daphnia is rather small. When the nutrition is rich, the head is also larger. So Daphnia grown in a rich nutrient environment shows a large head, right? Now what Walter Eck does is to continue to take it through several life cycles in this rich nutrition environment. So there is no surprise to find that you get large heads. The surprise is that at some stage, even when you remove the rich nutrition, the head continues to be large.
And now we say that there is some, so it has been incorporated into the.
Something
genetic.
has been incorporated, yes, yes, yes.
So this was just like when I was thinking about it, just a curious question that came to my mind, that how is it different from what the theory of acquired characters that Lamarck used in his theory?
Exactly. So I'll come to that. I haven't come to that yet. That explanation was given by Waddington. I'll mention it in a moment.
Ah, okay. Thank you.
The explanation was given by him in a moment. Okay. So this is simply to show a cartoon to tell you that many people since then have shown that it's possible to re-channelize development in different directions. I just illustrate one particular experiment done by Sondhi and Maynard Smith. Very elegant experiment too. And what they looked at were these bristles on the head segment of the fly, Drosophila. This was not Melanogaster, but that's a different matter. There are certain prominent body marks on the head sector, and 3 of those marks consist of these so-called false eyes or ocelli. 6 of the marks consist of bristles or hairs associated with these ocelli, and you see the picture on the left, the 3 eye spots and the 6 bristles. This is the so-called normal fly or wild type fly. Now it turns out that in the normal population, so-called normal population, so you have to use normal within inverted commas, this is the fly which is caught out there in the wild. Sometimes, very occasionally, you find flies missing an eye spot or missing a bristle. In fact, it happens that these features are correlated. Sometimes if it misses an eye spot, misses an ocellus, the bristle which usually accompanies that ocellus is also missing. Remember, the normal wild type has 3 ocelli and 6 bristles. What Maynard Smith and Sondhi did was to pick from their wild type collection, so this is a bunch of flies which you actually collect from the wild, so they're clearly members of the same group. From this wild type collection, they picked rare flies which had only 2 ocelli and 4 bristles. In fact, were missing the topmost ocellus and its pair of bristles. Picked 1 male and 1 female each of this kind. Mated them. saw what the offspring looked like. And most of the offspring in fact look like the wild type, right? 6 bristles, 3 ocelli. But some had, like the parents, 2 ocelli and 4 bristles. Now in the second round they mated brother and sister from these deviant types. Same kind of observation in the third generation. Most offspring look like the wild type, a few look like this aberrant type. But those few are proportionately higher than they were in the previous generation. So going from generation to generation, the proportion of offspring who have only 2 ocelli and 4 bristles gets higher, 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 higher. Ultimately it reaches 100%. And once it reaches 100%, there is no need to carry out selection any longer, because that has become the new normal type. So development in this fly, which was channelized in the direction of 3 ocelli and 6 bristles, has now become channelized in a new, completely new direction, which is 4 bristles and 2 ocelli. So this is once again a re-channelization of development carried out by selection, working on variation which pre-existed in this population. In this case, there was no need to change the environment or infest it. Same kind of thing that Volterek did, except that the source of the variation was there in the original population. It had not, the environment didn't have to supply it. The interesting part of this work as well was that they tried to channelize development in a number of directions, restricting themselves to this pattern, to the ocellular bristle pattern. Succeeded in every case in which the symmetry of the pattern was left undisturbed. The handedness of the pattern was left undisturbed. As you can see, the pattern is left-right symmetric, the way you see the sketch on the board. So for example, occasionally you get a variant form in which you have only the left, so now I am looking at the bottom pair of ocelli, where you have only the left ocellus and its bristle, the right ocellus and its bristle is missing. So in a sense, this is a left-handed fly as far as this trait is concerned. And they try to see, can we select for this left-handedness? Turns out that's not possible, because whenever you carry out or try to carry out such a selection, you do get flies which are in this sense left-handed, but you get an equal number of flies which are also right-handed. So at the population level, it appears that you can't select for asymmetry. I just mentioned that on the side to tell you that there are certain traits which may be so strongly canalized that they are essential for normal development per se and cannot be changed. Right. Waddington's experiment now. He also carried out a large number of experiments. His work mirrored that of Volterek in the sense that the changed phenotype that was now going to be canalized by him, or was eventually canalized by him, the changed phenotype came about due to something that he did. And in his case, what he did was a number of things in different experiments. So most often he used heat shock during embryonic development, or he used ether shock also during embryonic development, which is basically exposing these embryos to a vapor of ether. In both of these cases, he started with flies which were labeled wild type. So you pick them up from a bottle in your lab which is labeled wild type, because it consists of progeny from a population which was originally isolated from the wild and then maintained in the lab through successive generations of mass mating. So he picks flies from this population, mates them, exposes embryos either to ether shock or to heat shock. What happens? Well, what happens is that the embryos most of the time are killed, so nothing happens, no experiment. Very few embryos manage to survive but die as larvae, and even fewer embryos succeed in completing development all the way up to the adult stage, and even then they look sick in some sense, but they are there as adults. Fortunately, he found a few which were able not only to develop up to the adult stage, but become reproductively mature adults, and then he mated pairs of them. Same kind of thing that Sondhi and Maynard Smith did, except that this time it was done with an environmental insult, if you wish, supplied by Waddington. And the outcome, as you can guess now, was similar from generation to generation, each generation consisting of brother-sister meetings, exposure of the resulting embryos to ether, selecting from those few embryos which survived and which seemed to have an aberrant phenotype which was of interest to Waddington. In this case, what he was trying to look for was a phenotype which was known to be produced by a known mutation called bithorax. Ultra bithorax is the proper name, but the effect of that mutation was to give rise to flies with 4 wings. The pair of balancers or halteres, which are usually posterior to the wing, get converted to full-fledged wings in this mutant. So any fly which looked as if the haltere was larger than normal was picked upon by Waddington for these matings, and once again the observation was the same. After many generations, well, from generation to generation, the proportion of these flies which resembled the mutant bithorax fly kept going up. Eventually the proportion reached 100%. At that stage, there was no longer any need for the environmental shock. Once again, assimilation, and this is a word coined by Waddington, as I told you here, assimilation of a phenotype apparently into the genotype, because now it's breeding true, and apparently thereby demonstrating Lamarckian inheritance. Nonetheless, Waddington, who was a committed Darwinian, came up with a Darwinian explanation which subsequently was tested, well, it was partly tested by him too, but subsequently tested and shown to be held true. And this test relies on our understanding that what we call the wild type is only a type, meaning something on which you can put a rubber stamp and say this is genuine. You know, it's like this self-authentication you do when you have your Aadhaar card or something. It's that kind of authentication, but of the form, of the shape, of the kind of fly it looks like to you. That's what you mean by wild type. You implicitly assume that it must also mean a given defined genotype, but that's not true. Waddington pointed out that genotypes can vary a lot, but if natural selection has acted on narrowing down that variation, on increasing the signal to noise ratio, so to speak, that genotypic variation will somehow be suppressed. And the suppression Waddington suggested was by means of secondary genes, later to be called regulatory genes, which made these primary genes have activities which resulted in the phenotype flowing along the wild type channel. Presumably that happened simply because that was the fittest genotype. So natural selection led to that kind of outcome. What this environmental shock did, environmental perturbation did, Waddington suggested, was to expose the genotypic variation which was already there, because gene expression in general is perturbed by environmental shock, expose the genetic variation which is already there, and therefore de-channelize or at least reduce the strength of canalization of the wild type phenotype, increase the noise in the system remained the same, increase the noise in the system. But now by selecting on this variant phenotype, what Waddington was doing indirectly was selecting for those regulatory gene combinations which would make this new phenotype reliable. In other words, he was playing the same role that presumably, doing the same things that presumably would have taken place in nature had the fittest genotype been this genotype with 4 wings, genotype which gave rise to 4 wings. Is that explanation clear? So, perfectly conventional Darwinian explanation by which natural selection in nature, in this case artificial selection in the lab, actually selects for gene combinations which are not responsible for the phenotype per se, but are responsible for the reliability of that phenotype, responsible for assuring that phenotype is good. So, this has led to our picture now, this and similar work has led to our picture now that one must think of genes in development as playing two roles. Both of those roles involve whatever genes do usually. So these are DNA molecules. Sometimes they code for proteins. Sometimes they act as places where proteins bind to either allow the transcription or suppress the transcription of other genes. Sometimes they act so as to act as blockers to transcription by changing their three-dimensional conformation, etc., etc. Whatever else that DNA molecules do, they do. So there's nothing new in that. But the new point here is that conceptually you should think of them both as in some way being agents towards a particular end, and secondly in terms of being agents toward ensuring that that end is reached reliably. So it's a reliability of development that is also under genetic control that is now being highlighted by this kind of work. Again, a small point of historical interest for those of you who are interested in history. This picture you will have seen. This is Waddington's so-called epigenetic landscape. So you have valleys. Actually, initially he speaks of river flow, but then he decided that the pictorial impression would be made better by a ball rolling down a valley, and there are alternatives for it to choose from, alternative channels. The right hand side of that picture is not reproduced equally often, but it shows what he imagines is below this landscape, which you see on the left, which is a whole number of genes and wires leading from those genes to places in the landscape by means of which the landscape can be raised or lowered. So a valley can rise high or go further down. What looks like the crest can go down and become a valley, and so on and so forth. But all of you know this. This picture was actually anticipated much earlier by Sewell Wright, better known as an evolutionary biologist or a population geneticist. It's interesting. Wright says the same things that Waddington did. He's actually more explicit than Waddington in many ways, but his imagery is not as effective. So you can see from this directly, whereas from Waddington's picture you can see the ball rolling, going down one valley, the other, and so on. Wright illustrated it in terms of gene activities directing development towards one direction or another, in one along one route or another, and other gene activities narrowing the range of variation that these primary gene activities would permit. Really the same thing, nothing new at all, but pictorially less effective, as I said. So that's Waddington, that's his explanation. I should end now. 2 or 3, yeah.
It shows like how much actually a picture makes a difference. I didn't know about the Sewell Wright thing at all, but this Waddington picture is so appealing. I've
Very
seen
much.
it reproduced even in context other than development.
Very
For
much.
example, in Piaget, in Margaret Boden's book on Jean Piaget about like childhood psychology, Piaget uses the same
Yes.
visual
Yes.
aphorism and
Yes,
so on.
yes.
So I'm just wondering, like, this is actually thought up by Waddington himself?
Yes.
I see. So he was a magnificent visualizer.
He was an artist too.
Right.
Yeah. Beg your pardon? No. Well, these drawings I can't vouch for, but the drawing in his book, now I forget which book I took this from. He initially chose a professional artist to make the drawings. So it's the frontispiece to one of his books. Then it went through various modifications. As you see, there are 3 books listed here. It first comes in an introduction to modern genetics, then modified a bit, and then modified further in the 3rd book. Anyway, yeah, interesting you bring up Piaget's name. Piaget was a child psychologist who pioneered well, many theoretical concepts which are now commonplace in child psychology. But for us, what's interesting about him is that he was also for a while a committed Lamarckian, and he was very taken up with these Voltairic experiments and with similar experiments done by a person called Kammerer. That's a different story, which seemed to show Lamarckian inheritance. And Piaget was taken up with them because, you know, cultural evolution is Lamarckian entirely. Almost everything depends on learning and transmission to whoever else via learning. So Piaget spent a fair amount of time trying to show that Kammerer was right, but he didn't get there. Okay, that's by the way. So I just end with a couple of striking experiments in development which really illustrate what we now recognize as central features of the development form. One is this whole notion of gradients, and to my mind, it's been nowhere shown as prettily as in these experiments carried out by Michael Locke on an insect, on a beetle called Rodneus. And the experiments are noteworthy for more than one reason. One, of course, for their compelling nature. Even visually, you can see immediately the kinds of things that they show. Secondly, for the fact that he did this in the University of the West Indies in Jamaica, and it's the only paper I know of which was published with that address. And the third thing is that his experimental materials were only this beetle which he went out and collected himself and a razor blade, nothing more. Presumably he used a lens, if not a microscope, to see these patterns carefully and make these drawings. So what are these patterns? These are horizontal lines on the back of this beetle, repeated segment to segment. The beetle has the left half and the right half, so there is a midline which you see as that thick line in each of them, each of these drawings. And these horizontal lines are basically stripes on the back, that's all, nothing else. You can recognize them by means of a thickening of the skin, really cuticle, not skin proper, in these places, and also by the patterns of secondary hairs that appear there, which are not of our concern here. Locke's experiments were to take his blade, cut out small regions of the skin, transplant them elsewhere, after of course having cut out the skin from the place where he was going to transplant it. But before doing the transplant, he did a number of things. Either he took a piece of skin, transplanted it at the same level, took a piece of skin, transplanted it after rotating it through 90 degrees, or after rotating it through 180 degrees, clockwise, anti-clockwise, you know, all these things that would occur to you immediately when you look at this kind of thing. And the outcome in the pattern that you saw is illustrated on the right hand side. Now, I am not going to go into a detailed discussion of how this outcome can be analyzed, but you can see immediately that one way to do that is in terms of some kind of graded difference from one high point, let us say at the anterior end of the pattern, to a low point in the posterior end. And of course, you also need some rules by means of which this perturbed gradient, which of course you do not see, because the experiments are done at a stage where the pattern has not developed before the molt, which gives rise to the pattern. So this hidden pattern, this cryptic pattern, in some way has some dynamics which gives you the kinds of outcomes that you see on the right hand side. And this is one of the earliest experiments which convincingly showed that there were gradients in development. I just mentioned this to make the point that the notion of gradient is really pretty old and pretty convincing. Okay, now I have a whole series of slides which I am glad to show I displayed yesterday, and these were to make the point that to understand many aspects of multicellular development, it helps to go back to cases in which multicellularity arises by the coming together of single cells. So I don't have to repeat those slides, I'll just sweep through them very rapidly. And I will just summarize all the slides that we've run through before I come to this, which is going to be the end of my talk. So what I said yesterday was that situations of multicellular development in which single cells come together and form a multicellular unit, these situations can be formally compared to situations in which you have a number of individual organisms coming together and forming a social group. The outcome of this coming together is in one case something like a multicellular body with differentiation. In the other case, it is a social group with division of labor, which you can also think of as differentiation. And I tried to suggest that there are experiments which show, in both cases, well, meaning depending on which case you look at, there are experiments which go to show that part of the explanation for this differentiation or division of labor that you see later, part of the explanation comes from pre-existing differences in these individual units, be they cells or organisms, and part of the explanation comes from interactions between units after they come together. Now, when I say part and part, what do I mean? Am I saying that you have a bit of this, then something else which adds a bit of the other thing? No. What I mean is that depending on the situation, it appears as if either explanation can serve as a principle which accounts for what you see finally. Meaning, if inherent differences to begin with are strong enough, yes, they will act as determinant differences and give you the final outcome. However, if those initial differences are not strong, then interactions between the component units, either the cells or the organisms, will serve to spontaneously generate differences, which again you recognize as division of labor or differentiation. So, this is the point that I was trying to make, and I was trying to suggest that maybe there is some merit in looking at normal, so-called normal multicellular development also from this point of view, namely by thinking in terms of the embryos, beg your pardon, of the cells that make up the multicellular embryo as being in constant dialogue with each other, during which they both assess each other's internal state and modify their own internal states depending on what cues they get. Really pretty much the language that all of you people have been using in your presentations, and at least the other talks over the last 2 days use, except that there the language is explicit and refer to particular genes, activators, inhibitors, autoregulators, etc. The specificity of course varies from situation to situation, but formally this is what is going on. Now, recently, by which I mean within the past couple of decades or so, very interesting idea has been thrown up by a person by the name of Kupiec, Jean-Jacques Kupiec, where he has tried to expand on this notion, the notion that I presented just now, expand on this notion to look at multicellular development, really I am speaking of differentiation in particular here, to look at differentiation in multicellular development as the outcome of a Darwinian selection. I think the phrase is unfortunate, but this is his phrase, as the outcome of Darwinian selection between the component cells of the embryo. So, what he is trying to look at here, what he is trying to model here is the following kind of picture. You have a number of cells, all these cells have the same genotype. So, we are speaking of normal multicellular development here. In principle, each of these cells is capable of a large number of phenotypes consistent with one of those many channels that we have been talking about, that Waddington talked about. So, each cell can in principle go along any of those channels. Of course, today this does not need to be restating following pluripotent stem cells and so on. So, we know that this is really true in point of fact. Okay. Now, what happens? So, now we have a number of cells, each one of them being capable in principle of doing a number of things, but interactions between these cells stabilize the outcome so that the actual endpoints reached by these cells are in fact these different endpoints that you actually see as outcomes of multicellular differentiation. So, a very simple sketch shown here, you have two cells which he labels A and A dash. They are capable of expressing a number of genes, and in fact they do express many genes in noisy fashion, but the interactions among these cells are such and the feedbacks among these cells are such that that cell capital A ultimately ends up expressing a set of genes which he calls genes corresponding to phenotype B, and capital A dash ends up expressing a set of genes which correspond only to genotype C. All the other genes that they were expressing earlier have now been suppressed, they are not being expressed. Okay. So, how should one react to this kind of a model? First of all, part of it simply says what we know to be true in multicellular development. There is nothing new in this. in the sense that we know now today that every cell has within it the entire genetic repertoire which is present in the organism as a whole and therefore present in every other cell. And therefore we also know that the subset of genes expressed in any given cells is only a tiny fraction of the total number of genes that are present within the embryo, and that different genes express different subsets, to a large extent complementary subsets, other of course than the ones that they need for things like respiration, for example, or maintaining cellular integrity and so on, in which case are the common genes. So that part of the explanation is pretty common. What is not that familiar is this notion of noise in the beginning and this exploration of gene space, so to speak, by every cell as being a necessary feature of multicellular development. This is entirely hypothetical. As it happens, hematopoietic cells, which are essentially the cells that populate our blood lineage, which have been known for a very long time to start life as stem cells, which are capable of going into one of several lineages, all ultimately becoming the hematopoietic system, red blood cells, white blood cells, platelets, what have you, cells of the immune system, a whole lot of them. That this is known, that these stem cells give rise to whole macrophages, whole variety of cells. But recent experiments have shown that in the transition to this differentiation from stem cells to one of these other cells, in fact you see this sort of thing. You see noisy gene expression at the beginning. In other words, no cell type specific gene expression, rather a promiscuous pattern of gene expression, which later condenses into one of a recognizably small number of sets. So there's something maybe worth following up in this model. I won't go any further on this line, but will end in fact, so these are pictures that you saw yesterday, end in fact by showing two quick slides before coming to the conclusion. Quick slide number 1. This is what I wanted to push home yesterday as well, which is genetic identity in multicellular development is the thing taken for granted, but simply because of the fact that we are all genetically clones, we should not be misled into thinking that that's a prerequisite for multicellular development, for normal multicellular development. So this slide and the next slide shows examples of artificially engineered mosaics where you have not only cells from two different genetic lineages, but cells from two different lineages that separated millions of years ago in evolutionary history. Nonetheless, when brought together as embryonic cells, these cells are able in some way to talk to each other, to communicate with each other, to go through whatever patterns of gene activation they do. So this is a chimera between a goat and a sheep. Obviously you see the chimera after it has been born. The cells were combined in the embryo as blastocysts. This is a chimera between a chick and a quail. Here the divergence time is even further, 30 odd million years ago, where once again you've got a mosaic adult outcome resulting, again making the same point. You need not have genetic identity among the cells of an embryo in order for that embryo to be normally patterned and to develop as an adult. Rather, you need those cells to be able to talk to each other. Quite obviously, you need them to respond to the same stimuli, to be able to adhere to each other appropriately, make the same movements and so on, which seem to be generic properties, not as closely as we thought, tied down to genes, and of course later to differentiate into body parts. Yes, please.
How does the immune system know? Because it is seeing two, proteins of two different species. Do these chimeras have autoimmune disorders or something?
Yeah, no, that's a very interesting question, and oddly enough, the answer to that has come from human chimeras, which sometimes have been recognized precisely because the self non-self rejection, expected self non-self rejection was not seen.
I see.
Okay. So, what happens in these chimeras is quite often the immune system itself is mosaic. You get the point?
Yes.
Yeah.
Yes. Thank you.
Right. So, I will end with this slide now, which is the current sort of null hypothesis, if you wish, which seems to serve in a broad sense to account for at least some features of both development and evolution. And the null hypothesis is that both of these phenomena, both development and evolution, involve entities that at any given time have had a past history, therefore have certain properties at the present time. But having those properties, they are capable of exhibiting, when brought together, a great many outcomes as a group, in one case as a multicellular group, in the other case as organisms which form part of a species. These outcomes, the range of these outcomes clearly is not reliable because there is a range, but can be made more and more reliable by selection, and this selection involves the incorporation of secondary genetic consequences precisely to ensure reliability. I hope the point I am trying to make is clear. So, you have a certain outcome of groups of units coming together. That outcome is highly plastic, is highly labile. However, selection acts on those outcomes. So, fitness is a central component of this idea. So, one of those outcomes is better, so to speak, than the others. But selection acts to sharpen that optimal outcome, not by changing the steps that led to the outcome, but by making them more reliable, so that alternative outcomes are excluded more and more. So, natural selection really acts on the best adapted phenotype, and then results in a genotype which reliably gives rise to the best adapted phenotype. So, I hope I have in this, I am afraid somewhat discursive fashion, tried to show you that there is some merit in trying to look at both development and evolution from a point of view which stresses cells or organisms as the basic entities which embody whatever happens in the changes that we are trying to explain, and tries to think of genes as obviously essential factors, but not so much as directive factors as, if you wish, permissive as well as regulatory factors. Thank you.
Questions, comments?
I have a question. It is related to multicellularity again. So, as you discussed yesterday, and now you are discussing about social interactions and all those things. So, I was just curious, what if we have 500 around cells of slime molds, dictyostelium, and then we starve them, and from there they will make a fruiting body, and they will able to sporulate also. Now we do other experiment which has thousands of cells of slime molds. So what could we observe from there? Either they will make a bigger fruiting body, or they have two fruiting body instead of like bigger one.
Very good point. Okay, two answers. First, you can actually carry out an experiment, fortunately with these slime molds, which involves small numbers. In fact, you need not go to 500, you can even go down to 20. That's the smallest number that we could achieve.
Okay.
And the question that you can ask many questions, but one that you can ask is, does this make development more noisy? Obviously, are fluctuations more significant in this case? Answer turns out to be yes, as illustrated in these slides. There are 4 slides here because they were outcomes compared to various models which you don't need to look at. Please take a look only at the lower right hand slide, which gives you proportion of spore cells on the y-axis, and on the x-axis the number of cells within the group, and that number, I think the smallest number we got was 20, as you see there. And on the right hand side, of course, you go to as high as you wish. As you can see from this slide, there is a clear trend that with increasing numbers of cells, these proportions become more and more stable.
Right.
In fact, you get saturation at around 80%. As the cell numbers are smaller, the proportions become less and less, less and less reliable. So the actual proportions turn out to show a variation which is significant. Other question that you raised. So the first answer is that development becomes noisy. Second question, do they still form spore and stalk? Very interestingly, the answer is that they do. So there is one rare case, and I think that's the world record so far, in which a fruiting body of 5 cells was seen, only one case. That had both spore and stalk. Okay? Now, if you believe in the modeling that accompanied this paper, you can try and extrapolate the model to the minimum number of cells that you can get, which is of course 2.
Right.
Right? Multicellularity, you need to have at least 2. And ask, where does the model point to? And the model shows that then you get 50% stalk and 50% spore. In other words, one stalk and one spore. No one has seen that, so I can't vouch for that. Nonetheless, the answer to your question, you do get normal development with very small numbers, but that development becomes very noisy.
Okay. Like what if we increase the numbers?
That's what I'm saying. So you can move towards the right hand side on the x-axis.
Yes.
You can see that the y-axis tends to flatten out, and the flattening in this particular experiment is at about 80%. So that as you move to very large numbers, you get what you might call a Gaussian distribution of proportions with a small variance.
Okay, so the graph will be Gaussian.
No. This graph will just be a straight line.
So like I was trying to relate it with the group behavior in ecology. So basically there if we look into the animal pack, so they are mostly in like mostly 10 to 14 number, not more than that, because they think like if more people are there, then the efficiency of the group will be reduced. So can we relate this thing with slime mold, spore and stalk formation?
Only in a very formal sense, and that's pretty much almost an intuitive comparison, which is to say that when numbers are small, any sort of probabilistic decision making will lead to an outcome that becomes less and less reliable.
Any other questions? Yeah.
So somewhere in your talk you mentioned that noise in phenotypic variation is inheritable. I mean, it can be inherited in some.
Noise can be, I may have been misheard.
So I mean.
No, certainly, if I happen to say that, I withdraw it right away.
Okay, I mean, so how does noise? I mean, noise in biology, like we tends to consider as I mean, generally it is considered to be as a nuisance. I mean, and we tend to see biological systems as more robust to noise. So, so how does noise come into evolution in this evolutionary picture? And does it have any advantage? Like trying to, let's say, I mean, does like some environmental like disturbance can canalize a particular I mean organism to like have certain fate, right? But let's say noise in the system act as a buffer to kind of avoid that and fight that environmental disturbance, so that it doesn't go in the wrong direction. I mean, just I'm thinking in this direction.
Okay,
yeah.
well, two sorts of answers. One, both in development and in evolution, noise is an inescapable feature of the living state. So it's not something that you can wish away. In development, noise can come at several levels: transcriptional noise, translational noise, binding factor, protein-protein interaction noise. So all of this is noise in the conventional physical sense, where molecular interactions and molecular collisions, right, are inherently stochastic. So that you can't escape from at all. Secondly, as a consequence of this, you can get noise at the cellular level using the kinds of cartoons that I illustrated here, and many speakers have illustrated before me, where you can have feedbacks, let's say 2 negative feedbacks. There was a student group, I forget the number now, which had cascading positive feedbacks and then 2 negative feedbacks. You can see immediately that with these 2 negative feedbacks, negative cross feedbacks, there are only 2 stable states possible, one in which one pathway is active and the second is inactive, and then in which the first is inactive and the second is active, and you can reach either of these 2 pathways, right, depending on where the initial choice was made.
Yeah, so the initial choice is
no, but I use the word choice,
okay.
This is probably unfortunate, based on a stochastic input.
Right.
Right?
Right.
So noise plays a role. In evolution, of course, Darwin recognized this even without knowing what the source was. Uncontrolled random variation of the phenotype, I repeat, of the phenotype, is central to the Darwinian picture of evolution. After Darwin, we realized that this is based on these uncontrolled random variations in the genotype which occur, which we call mutations. Now, mutations also occur due to a number of reasons. One of them is again straightforward chemistry, let's say pairing errors during replication. And the other can be external causes like radioactivity, right, or cosmic rays, who knows. Right. So, noise is inescapable. It's there with you whether you like it or not. Now, you can call it a nuisance, that's a different matter. And that indeed was the way in which one usually looked at things and thought that noise had no role to play at all.
Right.
But as we see in both of these, it does have a central role.
But do you think like can it play as a can it act as a buffer against like to protect the organism from changing due to environmental stress? Like,
see, I'll tell you one kind of thing. I don't know the answer in the developmental situation, but in ecology there are many interesting models of how evolution in either reliably varying environments or in fluctuating environments, right, might mold the system in a direction which is inherently variable, noisy in our language. So, there are situations in which unique, stable directions lead to lower reproductive fitness than outcomes in which variation is part of the story. And of course, the word trade-off was used by more than one person. So, in situations in which there are trade-offs, it might make sense to place your bets partly on one outcome and partly on the other than entirely on one or the other. So, there too noise can play a positive role.
okay, yes. Thank you.
So, okay, question.
So when you said that first the phenotypic change happens and the genetic factors come and act as a stabilizer, let's say there is some adaptation due to environmental stressor, how exactly does that conversion happen in the sense that is it like in terms of epigenetics some chromatin gets activated and the gene that we already had is getting, or is it some kind of de novo sequence getting added to the genome, or is it like mutation gets added to or should I view it such that It's not the other way around. In a random population, all sets of species are getting mutations, and whichever mutation resulted in the increased fitness is actually passing on its genes. So is it like from that way should I see, or some kind of metabolic or whatever adaptation is getting really coded into the genome?
You know, the second picture that you sketched of a random mutation and selection among different phenotypes which resulted from the mutation, from different mutations, let's say, that is the conventional picture. And there's no doubt that it's a valid picture in a great many cases. I'm not in the least trying to say that that picture breaks down. What I was trying to say is that there's an alternative picture in which you do not need primary genetic change at all. Primary genetic change at all. Changes in DNA sequences at all. Okay? No mutations. However, on account of the inherent noisiness of outcomes, all of which are consistent with the same genotype, on account of that, you can select for alternative outcomes which were earlier unlikely but now become likely, and that selection acts via genes, right, which in this instance only serve to make that outcome more reliable, not to change the outcome. So to explicitly address the picture that you suggested earlier, you could have chromatin opening up non-specifically across the genome, making many different genes available for transcription, and all of this has happened, let's say, because of an environmental shock. But following successive cell divisions, it's possible that chromatin recondensation will shut off a great many of those genes and leave available for transcription only those which give rise to the phenotype which is optimal in this environment. Right?
With that, let's thank Professor Nanjana once more for his wonderful lecture. So before the formal end, let me just invite my co-organizers, Mithun and Shakti, once more here. So before I hand over the mic to them to say a few words, it's my pleasurable duty to thank the number of people who have been involved in successfully organizing this. So first and foremost, I'd like to thank our director, Professor Ravindran, who has always been very supportive of activities of this kind, our registrar, Srimati Vinyalata. But most of all, our super efficient administrative officer, Srimati Indra, who has effectively, like single-handedly taken care of almost everything we have asked her. So assisted by her office project assistant, Ms. Sadhana, whenever we had any issues, we just went to Srimati Indra and she promptly took care of it. Sri Johnson, whom some of you may have met because he's also in charge of the accommodation, was also taking care of the vehicles as well as, getting the registration kit together. So Mr. Johnson has been, essentially doing a lot of things to make this meeting possible. The reimbursements and the travel arrangements have been handled by Mr. Ahmed, Srimati Geeta, Sri Padmanabhan, and Sri Shankaran. Varuni has handled the social media publicity for this. Mr. Bhaskaran of the office has also been involved in, getting hold of the stationery. Then inside the auditorium, we had Srimati Gayatri and Sri Shiva Parimal, who's been handling the media, the live streaming and the recording. Mr. Darun and Bhuvanan, who are in charge of systems, Mr. Loganathan, Arvind and Ramesh for handling the electrical, ably led by Mr. Movindan. The catering was led by Sri Krishna Balaji, assisted by Mr. Rajkumar, and then The cooking team was led by Mr. Prasad and assisted by Atanu, Rakesh, and Mukesh. The catering serving has been done by Ravi Kumar, assisted by Raghu, Arun Deka, Karthik, Venkatesh, and Shravanan. The registration desk was handled by Mr. Johnson along with Manikandan, Sashikumar, and Moria Nikaja. Then the tea and coffee was served by Durganand and Avadesh. Now coming to our own team, so Nandini, and in association with Shaptoshi and Ashraf, has been like super efficient. In particular, Nandini has been, like working her butts off to make sure that all the TA forms are, submitted to the office right on time. So, she's been bugging you, I'm sure. So big hand to her. And a big hand also to the co-mentors Harish, Soling, and Shomadip, who you have had several encounters with. So Shakti is of course a co-organizer, but he's also been in charge of the web page design, assisted by Ashraf. And I would of course be amiss if I don't, thank my colleagues. Professor Rahul Siddharthan, who is right here in the audience and who's in charge of the IMSS Center for Disease Modeling and the Computational Biology project, which actually provided the bulk of the funding, as I mentioned. Big hand to Rahul. And my other colleagues, Arijit Samal, and Arijit Samal is of course a speaker in here, but also Sandeep has been coming regularly and has been always very supportive. And last but not the least, of course, to thank the Department of Atomic Energy for their generous funding, which makes all this possible. And I would now hand over the mic to my co-organizers, Shakti and Mithun, to say a few words before I make a couple of housekeeping announcements.
Thank you. So, as you've heard, this conference would not have been, or the workshop would not have been possible without the, the effort of so many people. In fact, it's taken so much of a burden of our shoulders. When I first sort of realized that this workshop is going to happen, I knew the final list of speakers, I felt this is going to be a fantastic experience. But at the same time, I was also quite worried because I really wanted this to go off without a hitch. And surprisingly, there has been absolutely no issues whatsoever, at least from my part. If you have any issues, if you have experienced anything, we would be glad to hear from you. But I am delighted with how this workshop has gone. So he has already thanked most of the people. Maybe I should just reiterate the immense support from the volunteers. The co-mentors have been extremely helpful in guiding the groups. It would have been a bit more of a task had the three of us just been working by ourselves. So the three co-mentors have been extremely helpful. Of course, Nandini and Saptarshi have been working so hard from day one. Ashraf, he helped me in the beginning to design the website. If not for him, there would basically be no website because I don't really have much skills in that department. And he also was very helpful in helping to post the to post the posters to the various institutes, which hopefully some of you have seen it in your host institutes. That's basically it. I mean, at the end, I would just like to thank all of you because the group projects have been an integral part of this workshop, and I'm so impressed by the quality of the presentations, and also the how much most of you have been interacting with all of us during the workshop, interacting with the speakers. You've been an extremely sort of receptive and sort of inquiring group of students, and hopefully this has been a great experience for you. I'd like to thank you. I'd like to and I would hope that that this workshop has sort of sort of inspired you for your future work. So with that, I'd like to give it to Mithun.
Thank you, Shakti. So I think I would be amiss if I did not take this opportunity to say that, I've been a co-organizer in name only. Shitabhra and Shakti are the people who have really put together this workshop, and they deserve a huge round of applause for all the hard work that has gone. So just a few final comments. I hope all of you have really enjoyed this workshop. It's really nice that at the end of these 2 weeks, which seem really long, but also really short, it seems like it's just started and it's gotten over, but it's also like we've learned so much. I hope it has been a really nice experience for all of you. I hope you've made friends. I hope you've learned a lot of science, and I hope, you go on to do great work in the future. These sort of meetings where, we spend a lot of time, mostly, nowadays we have 2-day conferences, 3-day conferences. That is a different experience, of course. We learn a lot. But these sort of smaller meetings where a group of us spend time together, learn science together, think together, these have a unique place. And I think Math Science, in that sense, has a tradition of these sort of meetings. So thanks to Math Science and its whole infrastructure as well. And I hope, I look forward to great science from all of you in the future. Thank you.
Thanks, Mithun. So a few housekeeping announcements. So I understand several of you are staying over the weekend. So let me just tell you that the food arrangement, as in the previous weekend, is only at the main campus. So if you'd like to take food, you can use the regular shuttle which starts at 8:30 from